Hello, my lovely anatomists and physiologists. Welcome back to our last video on the inner ear. The inner ear, remember, consists of three structures, right? So we have the cochlea, which looks a little bit like a snail shell. We have the vestibule, and then we have our semicircular canals. And remember, your semicircular canals are sticking up in three different directions. And remember, the bony labyrinth is surrounding the membranous labyrinth. There is perilymph fluid in between the bony and the membranous labyrinth. And then there is endolymph fluid inside of the membranous labyrinth. And that's going to be the fluid that we'll see involved in the sensation of rotation. So cochlea is where we're getting hearing. The vestibule is where we're getting our gravity and acceleration. And the semicircular canals is where we get rotation. When we're looking at our diagram from our OpenStax textbook, we can see that at the base of each of your semicircular canals, you have a set of structures which are called ampullae. So the ampullae with the AE ending is plural for the word ampulla. And this structure, remember, is housed inside the membranous labyrinth. And so we would have endolymph here. Notice you have those same hair cells as our receptor cells. We have the stereocilia, and again, those kinocilia with these cells. The stereocilia and kinocilia are embedded in a more gelatinous material called the cupula. And then the hair cells are also synapsing onto sensory nerves. So essentially what we see happening is as we rotate our head, the endolymph fluid swishes around in that semicircular canal in those different directions. And so that fluid is going to push the cupula. And when the cupula is pushed, then those stereocilia and kinocilia will bend. And this is going to send a signal on to the sensory neuron. If we go ahead up to our chart that we've been filling in, when we look at rotation, I think here I'm going to say the ampullae of the semicircular canals. And remember, that's in the inner ear. And then we have that same hair cell that we had in hearing and gravity and acceleration. Yes, gravity and acceleration. I think maybe let's make one more notation up here for hearing. That hair cell lacks the kinocilia. And each cell only has one kinocilium, and so we would call that a kinocilium as the singular version. Let's go ahead and map out for ourselves the rotation pathway. So in this case, I would just start with the rotation of the head is going to move endolymph. And when the, and let's go ahead and say in the semicircular canal, so we have that piece of information. And then this is going to, I'm going to say push the cupula. And when the cupula is pushed, then this is going to bend the kinocilium of each cell and stereocilia. So each hair cell has, of course, a single kinocilium and multiple stereocilia of the hair cells. And depending on whether they're being pushed toward the kinocilium or away from the kinocilium, we're going to see that the hair cell can be either depolarized or hyperpolarized. Remember, when we say a cell is depolarized, that means it's becoming more positive. So that's a stimulation. When a cell is hyperpolarized, that's becoming more negative. And so that's an inhibition. And so we're going to send the signal onto the sensory neurons, which are going to be making up the portion of the vestibulocochlear nerve. And the vestibulocochlear nerve, we know, goes into the brain stem. And then we're going to see, again, we have two pathways. So this kind of information can go straight to the cerebellum. Remember, this is that part of the brain that's doing the unconscious control of our skeletal muscles, smoothing out our movements, maintaining posture, regulating muscle memory types of movements. And then we also see this signal go on to the thalamus. And then, of course, it's going to go to the cerebrum for conscious level processing. Okay, that's it for the inner ear. Stay tuned for the eye. And as always, take care of yourselves and each other.